Hello everyone, welcome to lab tutorial. Today we discuss about erythropoiesis. The process of origin, development and maturation of a red blood cell is called as erythropoiesis. The formation of a blood, development and maturation of cell is known as erythropoiesis. Hematopoiesis means the process of origin, development and maturation of all blood cell is called as hematopoiesis. एरिथ्रोपोइसिस में आरबीसी का फॉर्मेशन होता है. हेमाटोपोइसिस में ऑल द ब्लड सेल्स इंक्लूडिंग आरबीसी, डब्ल्यूबीसी एंड platelets is known as hematopoiesis. What is the definition of blood? Blood is a special type of fluid connective tissue which is derived from the mesoderm. Hematology means the branch of a science which is concerned with the study of blood, blood forming tissue and the disorder associated with them is called as hematology. So overall the topic is erythropoiesis, which means it is clearly based on RBC and RBC formation, how to cell formation in our body, that's related. Then the process of erythropoiesis. The first stage of erythropoiesis belongs to the pluripotent hematopoietic stem cell. Pluripotent hematopoietic stem cell is further produce stem cell, and the stem cell is the origin of all the blood cell. Stem cell is always self-renewable, and it is also committed to produce different type of blood cell. The first stage of erythropoiesis is known as pro-erythroblast. Second is the early erythroblast. Third is the intermediate normoblast. Fourth is the late normoblast. Fifth is the reticulocyte, and sixth is the mature RBC. Reticulocyte is also known as immature RBC, which having reticulin fiber. Then the stage of erythropoiesis we already learned, that is pro-erythroblast, early erythroblast, intermediate normoblast, late normoblast, reticulocyte and RBC. Then go to the pro-erythroblast. It is the first cell derived from the colony forming unit E. Colony forming unit E से pro-erythroblast is first formation होता है. The size about 20 micrometer in diameter. Cytoplasm is basophilic in nature. It does not contain hemoglobin, that means hemoglobin are not produced in pro-erythroblast. Nucleus has nuclei and reticulin network. Then the next stage is early erythroblast. It is smaller than the pro-erythroblast and having the size is 15 micrometer in diameter. Cytoplasm is basophilic in nature, nuclei is disappear, and hemoglobin accumulation is started in this stage. That means hemoglobin formation होने के लिए जो सब essential substance required, those are starting formation in early erythroblast. Then the next stage is intermediate normoblast. Intermediate normoblast is the smaller than the pro-erythroblast, about 10 to 20 micrometer in diameter. Nucleus is still present. Hemoglobin is start appearing. Cytoplasm is basophilic due to the presence of a hemoglobin. It stain both acidic and basic dye. That means hemoglobin is start appearing in the intermediate normoblast, and hemoglobin is accumulation by early erythroblast. Then the next stage is late normoblast. Late normoblast is smaller than the intermediate normoblast. It is very small nucleus with condensed chromatin, having about size is 8 to 10 micrometer in diameter. Hemoglobin quantity is also increased. Cytoplasm is acidophilic in nature. That means hemoglobin quantity is also increased in late normoblast because the previous stage of intermediate normoblast hemoglobin is already fermented. Then the next stage is reticulocyte. Reticulocyte is slightly larger than the RBC. It has reticular network. Cytoplasm contain also ribosomal RNA. It is a non-nucleated cell. They are stainable with basic dye like brilliant crescent blue and a new methylene blue, which is also known as supravital staining procedure. Then the next stage in erythropoiesis is known as RBC. It is a mature RBC, about 7.2 micrometer in diameter. Reticular network disappear. Cell became biconcave in shape. Nucleus is absent. Hemoglobin is present, and the lifespan of RBC is 120 days. The total period required for the erythropoiesis is about 7 days. In this process, the different type of erythropoietic cells are formed in our body. Then go to the next step. What are the factors necessary for erythropoiesis? Erythropoiesis फॉर्मेशन होने के लिए कौन सा वो फैक्टर है जो बॉडी में रिक्वायर्ड होता है? The first one is the hormones part, that is known as erythropoietin, which is formed by kidney. Then vitamin B12, folic acid, iron, protein, vitamin C, intrinsic factor of a castor, growth hormone, copper, cobalt, nickel, and calcium. These are the essential substances which are required for the development of RBC. Then the next topic is properties of a blood. What are the basic properties of a RBC? The RBC color is bright red in artery and a dark red in vein. The pH is slightly alkaline. It is about 7.35 to 7.45. Taste is almost salty. Volume, it is around 5 to 6 liter in your body. Specific gravity is 1.0621. Mass is about 8% of the body mass. Temperature including 37 degrees Celsius of our body normal temperature. Viscosity is 3 to 4 times more viscous than water. So these are the properties of a blood which are important for our competitive exam. Then the composition of a blood, if we centrifuged a anticoagulated blood, then we got two different components. That one is plasma or a liquid part and the cellular element or a solid part. If you know about the cellular element, that is 45% having RBC, WBC and the platelet, and the plasma 55% contain mainly three basic component, that is known as water is regarding 91.5%, then the protein is 7% and the other solid is 1.5%. If we discuss about the protein 7%, then we get three important protein, plasma protein. Those are albumin, globulin and a fibrinogen. And the other solute having electrolyte ion, gases, nutrient and water product. Albumin is formation in liver. Globulin is also synthesis in liver. Fibrinogen is also synthesis in your liver. Fibrinogen is a clotting factor. Globulin having three to four different type of chain, that is known as alpha, beta, gamma and delta. The gamma globulin is produce antibody in your body. Electrolyte ions having sodium, potassium, calcium, magnesium. Similarly, the gases contain oxygen, carbon dioxide, nitrogen, and the nutrient part like our vitamins and other substances. Water product like urea, ammonia. These are the essential substances which are mainly present in our blood. So it is all over the composition of a blood which we get in if we centrifuged a anticoagulated blood. Thank you so much. If you like my video, then like, share and subscribe to my channel. Thank you.